lecture by Dr. Deepak Singh Bisht. He is my close friend. So Dr. Deepak Singh Bisht he's from Uttarakhand. He has done his graduation from Uttarakhand and post-graduation from GB Pant University of Technology, Pantnagar. He has done his MSc in biochemistry, and after that he has done his PhD in molecular biology and biotechnology discipline from IRI. And during his PhD, he has done work in Indian mustard regarding cytoplasmic male sterile line characterization and gene identification related to CMS line. And after his PhD, he has moved from brassica to rice, and now he is working on sheath blight, which is one of the most important disease of rice. And he is using genomics and transcriptomics approach for sheath blight gene identification related to sheath blight resistance, and he has identified some genes, and now he is applying this genome editing technology for sheath blight. So now he is giving a talk. He is going to give talk on recent development in CRISPR variant. So I invite Dr. Deepak Singh Bisht to give a talk. Thank you.
Thank you. And thank you, Anshul. Thank you for this wonderful introduction. So good afternoon to all of you. इसे चेंज करने के लिए अच्छा ठीक है, ये पॉइंटर है, ये चेंज करने आगे जाने के लिए ना. So as Anshul told, I'll be delivering a lecture on recent developments in CRISPR variants. I hope that by now, because this training has started for a couple of days from now, so you are quite you know many things about CRISPR, what CRISPR really is, how it was discovered, what is the role of CRISPR, how it can be used, how it can be modulated, all those things. Although my topic is specifically on these variants, but I'll just give you a brief idea so that you can feel the need of why we need these variants, why variants are exactly needed. Anyway, the thing can be done by Cas9 also, but still we need some variants so that the toolbox, this CRISPR toolbox can further be increased so that we can target different genes and different targets easily. Okay, so I'll give you a brief idea again about CRISPR. I'm sorry for the repetition, but then again it is needed here, so that's why. So the most important thing is that what do we need for editing? What specifically we need for editing? See, we will have a candid conversation so that you can gain something from this presentation, and also I can also gain from you. Okay, so a two-way conversation is very important for a particular lecture. So what is important for what do we need for editing? What is the foremost thing we need for editing? Yeah, we need to introduce breaks in your DNA. So it is not like this Cas9 is the only molecule that can induce double-stranded break in your DNA. There are other molecules also which are known for a quite long time that they introduce double-stranded breaks in your DNA. For instance, these are meganucleases, zinc finger nucleases, and talon coupled with this fork-1 endonucleases. So all these technologies are very robust, and they have been proven to edit n number of genes. There is a very famous example in wheat where genes present in 3 different chromosomes were targeted by talon. It was even the first paper in plants, in plant sciences, wherein this talon was used to target site-specific gene in case of wheat. Okay, but the important thing is that these technologies are very complex in design. So it is very challenging to design these molecules to target your specific gene of interest. And the advent of CRISPR has revolutionized this field because it is a very facile technology. It means that it's very simple to design. So anyway, whatever method you go, the concept is you have to introduce what? Double-stranded breaks in your DNA. Okay, so the fate of double-stranded break can be in 2 directions. The first one is it can go for non-homologous end joining. So this, and the other one is homology-directed repair. So this case, non-homologous end joining is an, am I audible without this mic also? Okay. So non-homologous end joining is basically what? It is a mechanism, okay, by which non-homologous strands can join, but it is again very error prone. So by this mechanism, there might be insertion of some insertions or deletion, there might be some disruption also. Okay. So but for this For this homology directed repair, what we need? We need a template strand so that it can go and insert into the site where the double-stranded break has been introduced. Okay, so this is the foremost thing, what is needed for disruption of your gene, for targeting your genes. So we need double-stranded breaks. Okay. Now CRISPR, all you know, CRISPR was first identified in what? Bacteria. So it is again a gift from nature. So this is basically adaptive immunity system in bacteria. So how it goes like, whenever there is an infection of phage in bacteria, the phage will, some small DNA pieces or some small chunks of DNA will be introduced into this CRISPR locus of bacteria. Okay, so in this CRISPR locus, they are now called as spacer sequences. And from these spacer sequences, these spacer sequences act as a memory signature. Okay, so when and the next time, when then again there is a phage infection, so this CRISPR RNA will be produced from this spacer sequences, and that will guide your Cas9 to the target. Okay, and then it prevents the infection of that phage into the bacteria. So on the basis of this is how this all system works, and on the basis of this, CRISPR system has been categorized into 2 different types. Broadly, although it is still a dubious classification, but you can classify this CRISPR system into class 1 and class 2 type. You know what is class 1 system and class 2 systems? Yeah, what? Class 1 are? Yeah, multiple factors are required for editing, apart from that Cas9. Okay, and in class 2? Single Cas protein can guide your CRISPR Cas9 system to your target locus. Okay, so that's why, so class 2 system is more easy to handle as comparable to class 1 system. That's why we go with class 2 system. And in class 1 system are type 1 and type 3, and class 2 systems are type 2, type 4 and type 5, type 4, 5 and 6. Okay. So the thing is that this was known previously also, but the discovery, whenever a discovery happens, it goes from a series of observation. It is not like that one fine day you get up in the morning, you will discover something. It is not like that. Okay, so this was like way back in 1970s, it was known that there is a presence of CRISPR locus. Okay, but it was not known what is the exact role of those CRISPR locus. And finally, when the genome sequencing project was, these are some observations that led to the final discovery. Then when this human genome sequencing project came into picture, there were several bacteria and archaeas were also sequenced. Okay. So during that sequence, they found that this CRISPR locus have these spacer sequences that are of viral origin. So it means that they might be having some role. So from that point, they came to know it might act as a memory signature. It was way back in 2005. And finally, it was found that these CRISPR locus contain an adjacent nuclease also. That nuclease is Cas9 nuclease. So from that point, the idea came that this might be helping bacteria in some immune response kind of thing. Okay, so this was the instance. So these are the series of observations which led to the finding that which led to this finding. Who are these people? You know, any one of you? And then? And Donna. Okay, so they are the one. And who else? Anyone else who is very related to these discoveries? Broad Institute. What is the name? Zhang. Yeah. So although there was a patent battle, this is beyond the scope of this discussion. But they were the first one. They published this paper in Science, and they first time saw that CRISPR RNA and your trans-acting CRISPR RNA can be fused into a single guide RNA, and that single guide RNA can direct your Cas9 nuclease to your target site. Okay, so this was the first paper, and this was the first discovery. Since then, this field has totally revolutionized the field of plant sciences. Okay, so this is how it looks. You have this Cas9 nucleases, you have this CRISPR locus, you require a trans-acting CRISPR RNA that will help in processing of your pre-CRISPR RNA into CRISPR RNA. Finally, that CRISPR RNA will guide your Cas9 protein to your target sequence and that leads into introduction of this double-stranded breaks into your DNA. Okay. Now, why this system? What makes it an ideal system for genome editing? Why it is an ideal system for genome editing? Specific can be done by talents also, and folk one that meganucleus also, but yeah, it is very easier to design. You have to only design what? That guide RNA, and theoretically by that guide RNA you can target any gene in your plant or your animal cell. Okay, So that is the beauty of this system. You need only a guide RNA that binds to your target or not, in this case, that binds to your target, and it requires only a PAM sequence. PAM sequence is protospacer adjacent motif. Okay, and that guides this entire system into the target, that Cas9 and this CRISPR RNA, guide RNA will bind to your target, and it will lead into direction of your double-stranded breaks. Okay. So there are two important Cas9 that has been isolated from Streptococcus pyogenes and Staphylococcus aureus. Okay, so they are routinely used. I'll tell you subsequently why these systems are. So now we'll gradually proceed towards variants. Okay. So what are the challenges with this CRISPR system? What are the challenges with this Cas9, CRISPR Cas9? First challenge is PAM requirement. You need a PAM like that juxtaposed to your target. Without PAM, you won't be able to target that particular gene. Okay. So you need a PAM sequence. Okay. So and that is a major challenge, because suppose a gene is not having a PAM sequence, then you won't be able to target it. Okay. Then again, off targets is a real problem, and sometimes these targets and sgRNAs are quite recalcitrant. Okay, then we are not able to target some specific genes due to some unknown reasons. Fine. So to address all these things, the need was felt to identify some CRISPR variants so that we can address these challenges. Okay. So the first challenge is that you can reduce the size of Cas9. Okay. By reducing the size of Cas9 means the size can be reduced so that it can be easily inserted into your cell. Sometimes you directly insert your Cas9 rather than making a stable insertion. Okay. Then increase the fidelity means you have to reduce the off target. Okay. So for this off target also, there are some variants now by which you can reduce these off targets. Finally, and then expansion of the scope of the target means if you expand the PAM requirements, you can the targets can be expanded. You can theoretically target any gene in your system. Okay, so this is the first instance where people tried to find out natural Cas9 variants. Natural Cas9 variants means, as we know that this is ubiquitous in bacteria, this CRISPR locus and the CRISPR system is an ubiquitous system which is present in almost majority of the bacteria. So the interest was that we can isolate Cas9 from these different bacterial sources, and meanwhile in doing so we can identify some bacteria which we can identify some Cas9 which is smaller in size and have an altered PAM requirement. It was a natural thing which could come into mind. Okay. So by this, the various group, they work separately, independently. They have identified Cas9 from different bacterial sequences, and fortunately, they identified Cas9 which were smaller in size as comparable to your SP-Cas9. Okay, but the problem was that the stringent PAM requirement. They require a very stringent PAM. So that's why till now we don't use all these Cas9. We are primarily focusing on this Cas9 only. But once we have a Cas9 which is smaller in size and the PAM requirement is less stringent, then it will be more adaptable. Okay. But from this discovery, these all efforts, one important system has been identified, that is your CRISPR and what? CPF system. Do you know what is this? Do you know what is this? CRISPR Cas9 is fine to all of you. These are the variants. The efforts were done to identify variants which are smaller in size, okay, and have less stringent PAM requirement. But so far we don't have these systems now with us. Although the size has been reduced significantly, but you see PAM is very stringent. To find these kind of PAM sequence, it is very difficult to find these kind of PAM sequence in your genome. Okay, so that's why this Cas9 is still routinely used, SP-Cas9 routinely used. But now we have a system that is your CPF system, CRISPR CPF system. Okay, so CPF is again an endonuclease. It has been naturally isolated from this Prevotella and Francisella. Okay, so in this case, the PAM requirement is NTT. Okay, and the thing is that it won't generate your blunt end cut. It will generate your staggered end cut. Okay, and other thing is that normally what happens, the cuts are generated adjacent just 3 base pair upstream to your PAM sequence. However, in this case, the cuts will be generated 18 to 20 nucleotide away from your PAM sequences. Okay, so this is a very simple system. Another important thing is that this system requires only a single CRISPR RNA. No trans-acting CRISPR RNA is required. Only a single CRISPR RNA, and this single CRISPR RNA can work with the CPF system. So it is now demonstrated that a CPF from this Acidomonococcus and Lacanospiraceae bacteria has been used for editing human cell lines and with great efficacy. Okay, so this is one of the system which is now picking up and working in parallel to that Cas9 system. Okay. नहीं CPF Cas9 जैसा ही है. It is same as Cas9. The only thing is that spacer means हाँ सिर्फ CRRNA यूज कर रहे हैं बस. हाँ रिपीट्स नहीं लेते. हाँ नहीं रिपीट्स नहीं, उसमें CRRNA के साथ हम ट्रेसर सीक्वेंस लेते हैं जो पूरा इसको GRNA स्कैफोल्ड बोलते हैं हम पूरा. ठीक है, लेकिन एडिट आपको सिर्फ चेंज करना है GRNA में. इसमें आपको सिर्फ क्रिस्पर आरएनए चाहिए, आपको रिपीट्स कुछ नहीं चाहिए इसमें. जो आपका GRNA है ना 20 बेस पेयर का, आपका उसी से बाइंड कर जाएगा, प्लस कुछ एक्स्ट्रा सीक्वेंसेस हैं जो उसको CPF को गाइड करेंगे, बट उतना कॉम्प्लेक्स नहीं है एज कॉम्प्लेक्स एज Cas9. ठीक है. The only and the thing is that in this case you generate this staggered end cut, or in that case you generate blunt end cut. Okay. So this was the first system, which was first variant, which they found that it might be working together with Cas9 system. Okay. Otherwise, all these Cas9 systems are not very routinely used because of only this stringent PAM requirement. Okay. Now. If you then again in this case you have to you have to fuse two systems. I'll come to this second slide. You can relate with that second slide. Okay. So now the thing is that the other challenges was that you have to increase you reduce the off target, increase the fidelity of your system. Okay. So how it can be done? Any idea how it can be done? You might have gone through the lectures, you might have read also. Any idea how it can be done? How you can reduce the off-target effect? Just any idea, apart from this slide, just any idea what comes to your mind, you can speak it out. RNP complex is, we'll come to RNP complex also, but in RNP complex, we directly introduced this entire ribonucleoprotein complex into your cell. There is no stable integration. So that reduced off target, but I'm saying that you have to generate some variants of Cas9. By generating variants of Cas9, can you reduce the off target effects? Any idea how you can generate? So this is a very simple thing. You can also you will see that how simple it is. In this case, Cas9 introduced double stranded breaks. Okay, so in this case the Cas9 is mutated from, it has two domains. Cas9 have two domains, you know, two integrase domain, HNS domain, RUVC domain. Okay, if you mutate one, it will act as a nicase. Nicase means it will introduce only a single stranded break. Okay, so one nicase is here, introducing a single stranded break, other is here, introducing a single stranded break. So if you target a gene with two gRNAs that are targeted towards in an adjacent sequence, so what will happen? The probability of finding this kind of sequence in your genome will be very less. Are you getting my point? See, rather than targeting your gene by single gRNA and a Cas9, you target your gene by two gRNAs that are coupled with different Cas9. Okay, one with Cas9 that will coupled with Cas9 nicase. Okay, so इसमें क्या है? gRNA है. Okay, then you have this Cas9 nicase, gRNA and this Cas9 nicase, and you are targeting a single gene. So what will happen? The probability of finding this kind of the complementary sequence of this gRNA and that two adjacent to one another will be very less. एक का तो आपको 20 बेस पर आपको मिल जाता है इजीली कॉम्प्लीमेंट्री, लेकिन अगर आप दो ले रहे हैं, दो gRNA ले रहे हैं, उनका टारगेट एक जगह पे मिलना डिफिकल्ट है ना, एक जगह पे मिलना, प्रोबेबिलिटी कम होगी. ऐसा नहीं कि नहीं मिलेगा, लेकिन बहुत कम प्रोबेबिलिटी. सो इन दिस वे इट विल रिड्यूस द ऑफ टारगेट इफेक्ट. Okay, so by having these two nicases, in this case this will generate a cut here, this will generate a cut here. So by targeting this, by having this CRISPR nicase, you can increase the fidelity of your genome editing cassette. Okay.
introducing two guide RNAs, but still means off targets can be there, no? Because single guide RNA can also bind.
No, but it won't be functional because it will introduce only a single stranded break. That break will be easily repaired by this DNA repair mechanism. You required, for editing, you require double stranded break, then only editing will happen. If only single stranded break is there, your DNA repair mechanism will come into picture. It will repair the break by taking information from your complementary strand. Okay, so in this case, one break is generated here, one break is generated here. Okay, so you require two breaks. In point mutation, this Cas9 has been done. Cas9 को change किया गया है. एक Cas9 में RUVC domain को stop कर दिया, एक MHNH को stop कर दिया. Anyway, it is functioning like a Cas9 only. When this both will come together, so तब साथ एक जैसा काम कर गया ना, लेकिन दोनों different है. ठीक है. So because the probability of finding sites adjacent to one another in a genome is less, कम होगा ना. एक का साइट 20 बेस पर मिलना तो आसान है, पर 20 अगल बगल मिलना टफ है ना, मतलब रेयर होगा, तो इसलिए ऑफ टारगेट रिड्यूस हो जाएंगे आपके. ठीक है. सो दिस इज द फर्स्ट वेरिएंट दैट हाउ वी रिड्यूस दिस, हाउ वी कैन रिड्यूस दिस ऑफ टारगेट इफेक्ट बाय यूजिंग दिस निकेस वेरिएंट्स. ओके. नाउ सेकंड, व्हाट कैन बी डन? In the another case, we can use a dead Cas9. Dead Cas9 means in which both the domains are non-functional. Okay, means the nucleus domain are non-functional. Okay, so dead Cas9 and gRNA is fine, but to this dead Cas9 you can attach what? Fork1. Fork1 is a in a nucleus that works, that introduce double-stranded break into your genome when they are in a dimer state. Okay, so fork1 require dimerization. Without dimerization, they won't act. Okay, so you can attach the fork1 to this dead Cas9, a fork1 to this dead Cas9. So this will favor dimerization also. It will introduce your double-stranded break wherever this gRNA binds to your DNA. Okay, the principle is simple. Is it fine to all of you? So these are some initial variants of Cas9. You have this nicase, and then what is there? Dead Cas9. Okay.
Dead Cas9 transformed plants are also called as mutants because somewhere I read that they are going to have this Cas9 always.
See, if you are introducing your gene, if it is a stable transgenic, then it will be there. But suppose if you are using RNPs, ribonuclear proteins, okay, so then it is not a stable transgenic. Na? What it will do, it will go edit, and in subsequent generation it will be removed. Okay, but यहाँ पे तो आपका integration stable हो गया, तो वो तो transgene है. लेकिन transgene is not a problem in case of plant system. The reason is what? Because in subsequent generation by back crossing you can eliminate it. Plants में कर सकते हैं ना. अगर suppose एक copy है, एक locus पे है, मतलब एक diploid है, एक क्रोमोसोम में एक ट्रांसजीन है, एक में नहीं है, लेकिन हमें तो एक्सप्रेशन चाहिए, किसी में भी इंटीग्रेशन हो. एक बार एक्सप्रेशन हो एंड दैट एक्सप्रेशन गो एंड टारगेट योर जीन, सो एडिटिंग इज परमानेंट, बट देन यू कैन रिमूव दैट कंस्ट्रक्ट सब्सिक्वेंटली बाय बैक क्रॉसिंग, है ना. इन प्लांट्स इट इज फिजिबल. सो इन case of plants, transgene is not a problem here in case of CRISPR-Cas9 because editing is permanent. You can remove this this construct later on from your this insertion later on from your plant system in subsequent generations. Okay. So this is another thing. This is an altered Cas9. This has been developed by using molecular evolution. Although molecular evolution is a technique by you can evolve a protein in lab to function, or evolve a protein in lab to act on different substrate. So the concept of molecular evolution is beyond the scope of this discussion. But then again, by simple molecular evolution, this Cas9 has been engineered. Simple this FN Cas9 has been engineered for NGG PAM to YG PAM. Okay, so these kind of works are also going on by different techniques, by mutations, modulations, and the Cas9 can be engineered with altered PAM specificity. Okay. Same thing is that simple this SP-Cas9 has been engineered for becoming an HF Cas9, high fidelity Cas9. Nothing has been done here. Only thing is that they have evolved this Cas9 into different kinds of Cas9, and they check their efficiency in human cell lines. Okay, and from that point only, they identified a Cas9, SPHF-Cas9, which was found to be very efficient in editing and without, or we can say with minimal off-target effect. Okay, the only way to check the off-target is that you have to sequence your entire genome. Okay. So now these are some natural variants, that how these variants can be used for high fidelity, having altered PAM requirement. And now this dead Cas9 and this nickase has now modified for having some varied functions also. Okay, so as you all know, this Cas9 has this HNS domain and this what? RBC domain. Okay, so if you mutate one, it will become nickase. If you mutate both of them, it will become dead Cas9. Okay, so this only principle has been utilized for developing large number of variants, and you will find these variants are really very interesting. Okay, so this is a brief outline what we will be going to discuss. Here, this entire system has been engineered to for gene regulation, for epigenome modification, epigenome editing, for chromosome imaging, for base editing, for RNA editing, for this chromatin imaging, and all those things. We will discuss all these different variants individually in the coming slides. Okay. So this is the first one. What is interference? What is RNA interference? You had a wonderful discussion talk in the morning session. So what is interference? Means it is interfering on the transcription basically of an endogenous gene. Okay, RNA interferencing means if you are introducing something from outside, some that goes into some mechanism and interfering the transcription of some endogenous target. That is interference. Okay, so this is interference is basically knockdown kind of thing, not exactly knockout. Okay, so this is CRISPR has now been used as a CRISPR interference. Any one of you know what is CRISPR interference? How it can be? See, all these things are very simple. You have to just think how it can be done. So what is it? CRISPR interference. Any idea? Means CRISPR-Cas9 is used to just interfere into the in the expression, or CRISPR-Cas9 can be used to alter the expression of a gene. Okay, it can be overexpression, or it can be repression. Okay, so how it can be done? One of the easiest thing to visualize is that if you are this entire CRISPR-Cas9 cassette go and dead Cas9 coupled with this gRNA go and sits into your target. So because it is already present there, so what will happen? It will hinder the transcription, because it will not let the RNA polymerase bind to that particular site. So it is the easiest thing to imagine. Is it fine? If you target your CRISPR-Cas9 cassette, in this Cas9 is dead Cas9. You don't want to induce breaks in your DNA. Fine? You don't want to induce breaks in your DNA. You need only a Cas9. You need a what? gRNA. gRNA, suppose you target a gRNA to your RNA polymerase binding site. Okay, so what will happen? Because of the hindrance in binding, your polymerase won't bind there. So the gene won't be expressed. Okay, so this is your CRISPR interference. So the first thing is that when dCas9 alone, only dead Cas9 can reduce or alter the expression of your gene of interest. Okay, but suppose if you want to further increase the efficiency of this particular cassette, what can be done? You can attach the repressor with it. You know, there are repressor in your genome. There are enhancers also. Okay, activators also. So you can attach a repressor. Okay, so one of the most routinely used repressor is your crab box, KRAB. Okay, crab box repressor. So this is basically present. This is a 45 amino acid long protein, which is present in your zinc finger domain proteins. Okay, so it acts like a repressor. So that repressor is fused to what? This dead Cas9. So in this case, what is happening? What is happening? Yes? Dead Cas9 is binding to the site. Anyway, it is hindering the binding of your polymerase. But the activity of the repressor is further increasing the activity of this particular cassette. Okay, further repression is now added. One step more repression is added. So this ensures that the gene is completely repressed. Okay, so this Cas9 can be fused to a repressor domain such as CRAB to enhance repression. So repression is enhanced. Okay, so is it fine to all of you?
In this case, we are targeting promoter region.
Yeah.
In between the genes, exon.
No, in this case, you can target a promoter region because RNA polymerase will bind in your promoter region only. Yeah. Fine. And now what else you can expect from this? Like interference, you can expect what? Activation also. How it can be done? By enhancing the activity. Then how it can be done? Adding of activators to your Cas9. Okay, so it is very simple. If you add activator to your Cas9, so it will enhance the expression of your gene of interest. So it is something like, what is activation tagging and all those things? It is something like activation tagging. Okay, so basically what are you doing? You're enhancing your expression of that gene. So it is very useful when suppose there are several genes in your system which are very important for your cellular growth and development. Okay, so if you silence those genes, if you knock out those genes, you won't get any result, because the cell won't survive. Okay, so in that case, what we do, we normally overexpress such kind of genes. Okay, so that we can, is it fine to all of you? So that we can see the phenotype, what is the resulting phenotype by overexpression. So we can relate the function of that gene and that overexpressed phenotype. So same thing can be done. The genes which we cannot knock down, they can be checked by overexpression. Okay, so this is the dead Cas9 plus. This BP64 is a activator, a trans activator. Okay, that has been isolated from this herpes virus. Okay, and this 64 stands for this is actually VP16. It is being repeated 4 times, so that it becomes VP64. Okay, that simple protein has been randomly repeated 4 times. So this is a very strong activator in case of human cell lines. So if you add this with your dead Cas9, so you can increase the expression of your gene. Is it fine to all of you? Now, yeah, please. No, it depends what you are targeting. It depends what you are targeting. Okay, it might, see, the thing is that these are very simple version of the entire concept. When you are going for targeting something, you have to check each and everything, that where to target, where it will bind, where polymerase is binding, where enhancers are binding. So these all things has to be checked before. Okay. Yeah.
This activator and repressor, it is universal for all genes or no?
No. This will depend from different individual to individual. This is basically for human system. See, in plants we don't anything that has already been validated into human system, that comes to plants later on. Okay. So in this case, these all things are first demonstrated in human cell lines. Okay. So this is basically what? Activation of your gene of interest. Fine. Now these all things has been further modulated so that the activation is further enhanced. Okay, so how it can be done? In this case, only we are having a single this VP64. If we add multiple copies of this VP64, the expression is further enhanced. Okay, so for these we have different systems also. We have DCAS9 SAM system, DCAS9 Suntac system, DCAS9 VPR system. The only thing is that I'll give a brief idea. We won't go into detail of these systems. In this case, what has been done? This Cas9 is fused to this GCN4. GCN4 is a polypeptide. Okay. You know this ELISA and all those things, the concept you know, that antibody, anti-antibody, then you add a fluorescent tag. Same thing is being done here. You have this GCN4. GCN4 is a polypeptide, and antibodies has been generated again this GCN4. And this the small chain of this antibody, small chain of this antibody is now fused to this VP64. So what is there? Cas9 is fused to this polypeptide, and you have this antibody fused VP64. So in cell, what will happen? If you introduce these things from outside, it will go and bind to this system. Now it will further enhance the expression of your gene. Is it fine to all of you? No? You are increasing the copies of what? Enhancer. Okay. So if you increase the copy of enhancer, it will further enhance your gene of interest. Okay. Now the thing is that in this case, it is little bit complicated. You know what are aptamers? Any idea what are aptamers? Aptamers are basically RNA or protein molecules that bind to some specific sequence, proteins. Okay. So in this case, as I said, aptamers can be RNA or oligonucleotide or protein molecules. Okay. In this case, they have changed this gRNA, and gRNA forms 2 aptamers. Okay. So this case, it is called scaffold gRNA. So these are aptamer scaffolds. Okay. So these aptamers are some specific sequences. They are already known. So this is MS2 aptamer. MS2 aptamer. Okay. And this aptamer is known to bound this MS2 coat protein. Okay, so you know this protein is a cognate binding, this protein is having the cognate binding domain of this aptamer. So what will happen if you fuse this MC2 with different, in this case you have different this activators. If you fuse this MCP with different activators and you have a gRNA scaffold like this with aptamers, so it will bind to this. So likewise you can increase the number of activators in your the entire cassette, okay, and it will further increase your expression of your gene, enhance your expression of your gene, okay. So these are different systems how by which you can use this CRISPR activation, okay. Now this is a very interesting kind of thing. We have this multimodal CRISPR RNA, CRISPR interference and activation function. Okay, so why it is important? In a single cell, you can activate as well as you can repress the expression of a gene. How it can be done? It is very simple. Suppose this is a gene 1. You are targeting this gene 1 with a gRNA. Having this scRNA means sgRNA scaffold. Scaffold, as I told you, it is mentioned specifically for sgRNA having this aptamer sequence. So you have aptamer sequence which binds to this protein, and that protein is tagged to an activator. In other case, there is a gene 2, you have this sgRNA scaffold, you have this aptamer, it binds to a different protein that is having a repressor coupled with it. Is it fine to all of you? If we introduce both this system into a cell, so at the same time you can increase the expression of a gene and repress the expression of a gene. So, is it fine? So by this we can go for a multimodal CRISPR interference and activation function. So it is very interesting. See, otherwise it is not possible. At the same time, and that too target genes. Gene number 1 is being expressed and gene number 2 is being repressed. Okay. So what is the advantage of this CRISPR RNA interference and activation system as comparable to RNA interference system? This you have to tell me. What do you expect? Yeah, less off target, but then RNA interference you won't have, you won't expect off target also. Yeah, it is like simplicity in design. Yeah, very simple design. Only gRNA you need, and then you have dead Cas9 already available, coupled with activators also available. Only thing is you have designed what? gRNAs. Okay. Then high specificity. Okay, it is highly specific. RNAi is not so specific, but in this case you have high specificity. Okay. And then final is directly control indigenous gene expression at the transcription level and can act on both coding and non-coding sequences. Okay. Now this is another important variation of this CRISPR system, and perhaps the most important one so far what we have discussed. These are basically base editors. Okay. So base editor means what we discussed so far was like insertion, deletion, disruption, but in this case we are just changing a single base. So see, it is very important. Single base means nothing else will be like anything else won't be inserted or deleted from there. Only base will be edited. Okay. So he is the man who pioneered this technology. And a base editor means just from your knowledge from your books, you might know that deamination of cytosine results into what? Yeah, please be loud. Uracil. And then it will conversion is what? C to T. C to T. Okay. So you can change this C to T by just deaminating your cytosine. Okay. So from that concept, what is needed first? You need a deaminase. Is it fine? If you coupled a deaminase with your Cas9, if you fuse Cas9 with deaminase, it will go and edit your cytosine. Okay. So the simple thing is that you require a deaminase and you require this Cas9. Nothing else is required. gRNA for that particular target. And the editing will be done in a window of 4 to 5 base pair from your PAM sequence. Okay. So now you need a cytidine deaminase, you need a Cas9 protein. I'll come to this, why it is needed. So by this you can change, and in this case you need a what kind of Cas9? You need a Cas9, dead Cas9 or a complete Cas9 or nicase? You need a nicase here. I'll come to this point also why we need a nicase here. Okay. So you need a nicase. See this example. You are having a C. You have this entire machinery here, gRNA coupled with your deaminase and this uracil DNA glycosylase inhibitor UGI, it will result into conversion. C is now converted into T. So this is your base editing. Okay. So by this you can ideally change any cytosine in your living cell. Okay. Now why this is needed? Inhibitor. See, whenever there is editing like this, the DNA repair mechanism will come into picture. Okay, so what it will identify this U as a mismatch. So this inhibitor, this DNA glycosylase will come, it will remove this U and again put C there. So you require inhibitor also, so that it won't come there and it won't function there. Okay, so that's why a glycosylase inhibitor, a deaminase and Cas9 complex. Is it fine? Now we will go into more detail of this. So this is a simple example, nothing related to that, but in this case this is a CRISPR stop, it is called I-stop. I-stop means that this CCA, CAG, CGA, if you change this C into U, what will happen? A stop codon. Okay, so you can stop a gene or a protein prematurely. Okay, so now by this technology you can introduce stop codons into your gene also, and this technology, that premature stop codon is less deleterious as comparable to your complete knockdown. Although the gene is not functional here, but still it is less deleterious as comparable to your complete knockdown. Okay. I missed one slide actually. Okay, I'll conclude here only. So you have this glycosylase. So that glycosylase inhibitor means what? It will inhibit what? It will inhibit the removal of uracil. And now this U, you know how it happens? U will pair with what? A. You will pair with A, and now subsequently A will pair with T. So that's why this C is now converting into AT. Is it fine? Likewise, you have this adenine base editor also. Okay, I missed that slide from here. No way, we will proceed further. Fine. Now, apart from this base editing, what you have this? Epigenome editing. Okay. So, you know what is epigenome? What is epigenome? Yeah. See, it is written here. Epigenome describes all post-translational modification and other promoting features associated with regulatory element in the genome. Okay. The genome is missing from here, but you can put it here. Okay. In the genome. So, one of the famous methylation is what? Methylation pattern is DNA methylation. Okay. So, in plants there is having these methylation has different significance. However, in case of animal system, this you have large number of cytosine methylation. Okay, 5-methyl cytosine in the CG islands. Okay, they have a significant role in case of cancer and all those things. Okay, so you have this methylations in your in your genome, and these methylation play an important role in expression of your gene. They can repress also, they can activate also. Okay, so the important enzymes which regulate these methylations are DNA methyltransferase. This is methyltransferase 3A and 3B, and you have another maintenance methylase also, but that is not important here. So what we need, so if you wanted to change methylation of a particular cytosine, what you need only this enzyme, methyltransferase. If you couple this methyltransferase with your Cas9, what will happen? It will go and methylate your C. So this is your epigenome editing. Is it fine to all of you? Very simple, nothing to do. Okay, you have this DNA methyltransferase, it is coupled to your this Cas9 complex, and now this methyltransferase will go and target the C in the vicinity of your target locus, and will change a C into 5-methyl cytosine. Okay, and same way if you attach if you add this demethylation enzyme for what are the examples of this? 10-11 translocators, TET. 10-11 translocators are the enzymes which remove the methylation from your cytosine. So if you want to remove your methylation, what you will add? This TET enzyme, TET protein. If you add methylation, what you will add? DNA methyltransferase. Okay. So both things can be easily done from this these kind of modifications. So this is a basic construct how it can be done. You need a dat Cas9, you need a methyltransferase, and then this is an antibiotic for selection. Okay, nothing else is needed. So the design is also very simple. Fine. Now the final thing is that you know that when we go in when we do routine work in a lab or in a lab, we go for sometimes we feel the need of imaging. Chromosome imaging is normally done by fluorescent in situ hybridization. You might be knowing from your like bookish knowledge. You go for this FISH. Okay. The only challenge is here is that you need to like fix your tissues. You cannot go for live cell imaging. So for live cell imaging, this CRISPR has provided a very good alternate. Okay. In this case, what to do? You have to add this GFP with your Cas9. Is it fine? gRNA targeting the locus, and you have to add this GFP with your Cas9, and now you can see exactly where that gRNA is binding and see in microscope. So is it possible? Very easy, na? You target a GFP with your Cas9, and then you can see the pictures in your microscope. But the thing is that only one GFP won't be sufficient. At a particular target, how many gRNA can bind? Only one can bind. But the fluorescence emanating from this GFP won't be sufficient to be captured by even a very good high quality of microscope. Okay. So what to do? So the initial experiment for these kind of things were done in repetitive sequences. So if there are repeats in your genome, so multiple gRNA will bind. Now the signal will be sufficient to be captured by a microscope, so that can be easily captured. So this is very easy when you are imaging your telomere dynamics or some dynamics of some repetitive element in a live cell. Okay. So this can be easily done, but the problem is that, but suppose if you wanted to identify some genic region, a single copy gene, so how it can be done? Any idea how it can be done? See, I told you initially aptamers can be done. Okay, so same thing, you add this aptamer sequence with this guide RNA, gRNA scaffold, you have these proteins which bind to your aptamers. These proteins are tagged with your YFP or GFP, whatever you want. So now you increase the number of your GFP signals. Okay. So suppose if this protein, this entire gRNA Cas9 goes and bind to a target. So see how many YFPs are here. So that signal coming from this particular binding will be sufficient enough to be captured by your microscopic examinations. Okay. So by this way you can go. So these things were not possible previously. CRISPR-Cas9 has made it possible now. Okay.
Sir, this can be used for checking the transformation efficiency also. If we are not using the Cas9,
You can check it. Yes.
Cas9.
See, you can do n number of things by all these things, whatever you want. The only thing is that.
There is confusion that the plasmid is inserted or not, like that.
No, but then.
By doing the screening. So if we are using any kind of fluorescent marker,
you can check it.
Cas9,
You can check it. Yes, you can check it.
we can screen on
You
the
can check it.
dead
But in this case, you are having this dead Cas9. It won't be useful for your purpose because you need a proper Cas9. This is normal Cas9 will go and cut your DNA there.
But if we want to create the mutation also,
You want to create the mutation, no? But in this case, the purpose is not to create mutation. So if you add a normal Cas9 there,
if we design,
you design it, but then again, so in this case, because you need that Cas9 for mutation also. So if you have such kind of thing, it will go and create mutations there. So for imaging purpose only, it is fine. But I don't know whether it can be modulated for checking the transformation efficiency also. Because if something can be done, that you can add an other Cas9 or something, a close by sequence can be added, then it might be possible. But I don't have found like any such kind of examples. Okay. So this is fine to all of you. Okay. So you have this. By this you can increase the signal, and those signals can be easily captured. Okay. Now this is the final conclusion. But before that, any questions? Like, because see, I tried to make it very simple because I knew that you all are students, so that it might help in your academic endeavors also. So that's why, although I have few more slides also, but I won't show them here with some examples also. But then again, any questions? No questions? Okay. So these are some future directions. Okay.
Sir, to base editor system, the like a point mutation, so how to differentiate by the spontaneous point mutation or by the base editor system?
See, spontaneous means spontaneous. This is also a mutation. This is also a mutation. Okay. So spontaneous mutation happens during a course of time. It is not like that because you know that suppose you have targeted a region called X. Okay. In that region you have a C there, and now by your base editing that C has changed to A. A में बदल गया हो, ठीक है? And now you can sequence that region only. क्यों C में बदला है, C बदला कि नहीं बदला वो चेक करने के लिए. आपको अगर पूरे को जीनोम को कंपेयर करना, इट इज नॉट पॉसिबल. कि आपको कैसे पता स्पॉन्टेनियस म्यूटेशन का फ्रीक्वेंसी पता कितना होता है? 10⁻⁶. ठीक है, मतलब आपको इतने सालों में एक म्यूटेशन होता है, इनसे आप लिंक नहीं कर सकते इसको. दिस इज अ डिफरेंट थिंग. दिस इज ओनली फॉर अ टारगेट, एंड टारगेट यू नो, तो आप कुछ नहीं करो, बस उसको जहाँ पे एडिट हुआ सीक्वेंस कर लो. आपको तो पहले से पता है टारगेट तो ना, तो आप कंपेयर कर लो एडिट हुआ कि नहीं हुआ, यही आप कर सकते हो इसमें. लेकिन आप यू पिक्ड अ वेरी रेलेवेंट पॉइंट. द थिंग इज दैट दिस माइट रिजल्ट इनटू editing of different bases present elsewhere in your genome. Then it becomes very difficult how to identify. Although you are targeting a specific sequence, but there might be some mismatches. There might be some, this dead Cas9 might act on some other cytosine also. For that identification, you have to sequence your entire genome. Okay, so efficiency is still like debatable. You have to standardize these technologies for future applications. Okay. Now, these are some future directions. Like what we need more is we need a smaller Cas9, particularly for human system, and for this RNPs also, because if you have a smaller system, you can directly inject into your cell. Okay, then the other thing is that see, this is very important thing. These Cas9 are all from what? Bacteria. So it is expected that they will be immunogenic to human system. In plant it is fine, but in human it will be. But if in plant it is a stable transgenic, then it will be again a problem. Okay, it might be attract a debate on this, that whether it can be used or not. Then again go by all those bioefficacy methods of checking the immunogenicity of this Cas9 protein. So it is expected Cas9 is from bacteria, and now people are different people have different viewpoints. So it has already been proved that half of human may already have pre-existing humoral cell-mediated adaptive immune response to Cas proteins. So still all these technologies have a long way to go into the market. Okay. Now ethical issues are a major concern. See, whenever two people sit with a different viewpoint, you can always debate on issues. Okay, if you have a knowledge of that particular subject. So ethical concern will always be there. Okay. So these are some future things which are to be addressed, and hopefully in the coming time it might be addressed. Okay, I'll conclude my lecture here. I thank you for your patience hearing, and if any questions are there, feel free to ask.
Can you explain any example multimodal CRISPR function?
See, I have several examples of this, but I haven't put it here because these all systems are validated in animal cell lines. So, but that slide is not here. Explain means you want to explain how it was done. How it was done will be very difficult to explain, because then to how design those construct, how it was transferred, it will need a separate lecture on this. The only thing was here to give you a brief idea that these things are possible, so that when you go back to your respective places, you can search the literature for all these kind of findings. But I hope that you might have some idea from this, that these things are possible, these things can be done. Okay. Yes, what else? No. In plant cell also it can be done. In plant cell. See, I told you initially that all the discoveries, first they are validated in animal cell lines, because it is very easy to grow animal cell lines. You don't have plant cell lines. Animal cell line तो आप ऑफिस में लैब में ग्रो कर सकते हैं ना. प्लांट सेल लाइन तो ग्रो नहीं होता. इजीली इन्फेक्ट कर सकते हो आप, इंजेक्शन से उसमें इन्फेक्ट कर सकते हो, देख सकते हो उनमें. एंड लार्ज ह्यूज research is already been done in these animal cell lines. So it's very easy to demonstrate in it. Plants, for demonstration in plant, you need to develop stable transgenics. Okay. So it becomes difficult in plant. And then again, plant require a generation to grow. Animal तो आपका क्या है, कुछ दिन में RBC है, ल्यूकेमिया सेल लाइन्स है, हीला सेल लाइन्स है, वो कुछ दिन में ग्रो हो जाएंगी. आप चेक कर सकते हो ना. प्लांट सेल तो नहीं कर सकते ना. अगर आप किसी मैंगो में ग्रो ट्रांसजेनिक चेक करेंगे, तो मैंगो जब फल देगा तब तक तो हमारी पता नहीं सेकंड जनरेशन आ जाएगी चेक करने के लिए, है ना. तो चेक नहीं कर सकते हम लोग. और कुछ ideal length of, see, in this case we use a 45 amino acid. The length of the scrap is 45 amino acid. So see, it is very important question, because that these all thing has been standardized. It is not like that you add a big repressor and you introduce into a cell. Everything has been standardized. So in this case you have a 45 amino acid long protein, that crab repressor. Okay. So if no more questions, then I have a short quiz for you. You might solve it later on after leaving the class, after this lecture.
You can write these questions.
Now perhaps you can answer, only 3 of them are there. If the target does not contain a PAM sequence, will CRISPR-Cas complex bind? It varies basically, it varies. Okay. It doesn't have PAM, it doesn't mean that it won't bind. Okay. See, one of the important thing why I am saying this, see, I can relate this thing. Why? This CRISPR system is very generic, it's not so specific. Why this statement is there? Because that bacteria has to face every time some new pathogen, new phage. So if that system is very specific, so it will act against a single phage. So that's why that system is not so specific. It is somewhat liberal. It binds to some different sequence of different specificities also. So that's why we have this off target also. You're getting my point? इसी वजह से होता है क्योंकि बैक्टीरिया स्पेसिफिक अगर होगा ना तो वो एक ही के अगेंस्ट होगा. सबके बैक्टीरिया तो ऐसा तो नहीं है एक फाज इन्फेक्ट कर रहा है. During the course of time, many things will infect a bacteria. So bacteria का system ऐसा develop हुआ है कि it has the complexity is little bit reduced. It might act to some different classes of phages also. So that's why you have this off-target effect also. How often PAM sequences are present in mammalian genome in average? How often are the PAM sequences presented in the mammalian genome in average? ये आप लोगों को देखना है, ठीक है? If Cas9 is stably integrated into the genome, and is it continually acting on the genome and causing mutation even without the PAM, even without the gRNA? This also you have to see. Okay. Yeah, answer है. Okay, thank you.
So at last I want to thank Dr. Deepak Singh Bisht, who has given a wonderful talk on various variant of Cas9. So he started with the CPF1 system and then dead Cas9. Dead Cas9 is like it is attached with a activator and a repressor, and at the same time you can activate a gene, you can repress another gene. So then he has covered about base editor and specifically in detail about cytosine base editor and various epigenome editing, and at last he has covered a live cell chromatin imaging. So so I want to thank Dr. Deepak Singh Bisht and give a big round of applause.
Thank you.
So we want to give a